Thank you so much for coming out on this beautiful Southern California evening to hear about directed evolution. I'll try to make this worthwhile for you, but I want to thank Nancy and Elliot and the rest of the committee for having me here. I'm just your neighbor over in Pasadena, but I've never been to Mud before, and it's been really a lot of fun. And all you Muddites, please apply to Caltech for graduate school. We have a fantastic program in synthetic biology. Lots of professors. If you don't like me, you'll find somebody else you don't like. And we will really do well by you. This is such a cool field. And I love being an engineer in engineering biological systems. In fact, one of my really fun jobs as an engineer is serving as a judge for the new Queen Elizabeth Prize in Engineering. And I hope one of you will win this prize someday. It's for inventions that really change the way we think and that change the way we live. And the first prize was just given 2 years ago for the internet. So there's an engineering invention that really has affected the lives of billions of people. And I'm hoping that some of you will be on that roster in the coming years. But the QE Prize community, well, they engaged the engineering community recently on Twitter. I'm sure a couple of you are on there. I'm on Twitter, so please follow me. And because I'm interested in getting young people interested in engineering, and you might not even have thought how interesting engineering is until you hear what I do. But the Queen Elizabeth people said, okay, let's think of the best, the greatest engineer of all time. You know, we have to give the award to living people. That's kind of a constraint. But when you think of who is the greatest engineer of all time, is it Archimedes, Leonardo da Vinci? Is it Thomas Edison? Well, I didn't have to think more than a microsecond on this one. It was really easy for me because for me, the greatest engineer of all time, there's only one answer, and that's nature. Nature is truly the greatest engineer of all time. Because when you think of biological systems, nature has figured out how to extract materials and energy from the environment and convert that into self-replicating, self-repairing, adaptable, moving, sometimes even thinking machines, all with remarkable efficiency and elegance. And the products of almost 4 billion years of evolution are all finely tuned. This evolution, this exploration, this innovation, this trial and error, they're all finely tuned to solving a really important problem. Nature has solved a really important problem. What is that? That's being alive. The problem of being alive. Nature solved that really well. And not just being alive, but enduring for billions of years. Nature has done this with remarkable organisms, which of course are made up of even more remarkable molecular machines, and this is what I'm going to talk about tonight. How are these machines made, and how can we build new ones? But when you think about these machines, these convert one form of energy into motion. These catalyze all the reactions that build up life. These store information and read information from DNA. These convert energy into motion. All these machines are solving problems that are not unlike some of the problems that we humans have to solve in our bid to thrive on this planet. So we're astonished, in fact, by some of the solutions that nature has come up with. And of course, my chemical engineering background comes through on this. I'm astonished by how marvelous nature is as a chemist. These little molecules, these little organisms can do chemical reactions that human chemists have not figured out how to do. And they do them very beautifully and very efficiently. And if we could learn from these systems, I feel that human engineers have a great deal to learn from biology. And one way we can learn from biology is to learn how to exploit some of the 4 billion years of evolution in order to solve human problems. So, for example, these chemical systems, these efficient catalysts that nature has developed could help us, for example, in our bid to, something There. They could help us in our bid to utilize renewable resources. You know, you think about it, biological systems know how to extract carbon dioxide from the atmosphere and combine that with the sunlight and a little nitrogen from the ground in order to build new molecules and living systems. They know how to use waste materials. They know how to use things that are largely sustainable and renewable resources. So we can learn a lot from these systems, and we should be able to exploit these systems in solving human problems. So this is where synthetic biology comes in. All this functionality, and this is the remarkable thing about biology, all this functionality is encoded in a simple 4-letter code in DNA. So many of you are young. This was only discovered, the structure of this was only discovered when? 60 years ago. And since then, there's been this remarkable revolution in our ability to interpret DNA and to even modify it and to reprogram life. So that's what this new field of synthetic biology is about, is how do we reprogram life and hopefully use that to solve human problems. All right, here's the problem. Now, you've probably, since you are all cognoscenti in the synthetic biology field, you probably saw these headlines from 2010. Craig Venter's team synthesized the entire, from non-living material, the entire genome of a simple organism. I find it a very boring organism, but the entire genome, 1 million base pairs, they could write it out, synthesize it from non-living material. When they put that DNA back into these cells whose DNA had been destroyed, the cells read it as if it was their own. They said, oh, fine, and they rebooted. So they said, we rebooted the cells, but the cells read the DNA and they started reproducing as if nothing had happened to them. And they read the DNA. That was an amazing technological advance that goes to show and demonstrates beautifully that we can write out any sequence of code that we want. But here is the real challenge, and this is the challenge that I'm hoping that you, some of you in this room, will be able to solve, and that is, how do you compose DNA? Right? We know how to write it. We know how to synthesize it. You can synthesize it from non-living materials, but we don't know how to compose the sequence of letters that encodes an organism that converts carbon dioxide and sunlight into gasoline, for example. That is not something that nature figured out, because nature doesn't really care about your big gas-guzzling car. Nature likes to compose DNA that makes more organisms. It does it really well. But if you want to subvert natural systems to solve human problems, you have to rewrite this composing, you have to rewrite this DNA, and you have to recompose it. And to me, that composition is beautiful. It's intricate. It's a product of 4 billion years, and we don't know how to do that. We're just learning how to hold the pencil. So let's get real here. Let's get down to molecules. Let's just take what you might think is one of the simplest problems. Forget about writing the whole organism that makes a biofuel. Let's just make a single protein. We learned what the DNA code represents in terms of a polypeptide sequence many years ago. So we know this first step. How does DNA make a protein sequence? So that's a solved problem for the most part. But what's the second problem? How does the polypeptide chain fold into a well-defined three-dimensional structure, or not well-defined, but a three-dimensional structure? What's that problem called? It's called the protein folding problem. When I was a graduate student 30 years ago, I was told that problem would be solved in 5 years. Were any of you told that? Do they tell you that problem's going to be solved in 5 years? Well, they're lying, because it's a really complicated problem. It's a very difficult problem, and it's a problem that we don't yet know the answer to. But you know what? They don't even talk about that problem, because the more important problem, which is not how it folds into a three-dimensional structure, only biochemists care about that. Engineers care about what does it do, right? What does this molecule do? What is its function? And how does the DNA sequence encode that function? So you have these two great big question marks that we are a long ways away from solving, especially from a point of view of being able to use it to write out DNA that does something useful. So given that these problems are still major problems in the field, what is an engineer to do if you want to rewrite biology? Well, if you study biochemistry long enough, you'll realize that problem has been solved. There's a very simple algorithm, turn the crank type algorithm, that has given rise to all these beautiful functionalities, all the beautiful proteins that you can scrape off the bottom of your shoe. And they're all finely tuned for their biological functions. We know that proteins, from just looking at the sequences of proteins that exist today, we can put them into families where we know that they came from a common ancestor by accumulating mutations and going through the filter of natural selection, so that today, after hundreds of millions of years, we have a whole platter full of proteins that are beautifully tuned for biological function. And this design algorithm did not require you to sit down and compose the DNA. So how do you utilize evolution as a forward engineering algorithm? How can we redesign biological systems using evolution? Not a new idea. In fact, humans have been doing this for a very long time. We have been rewriting the biological world, synthetic biology, nothing new here. We have been modifying the DNA and the biological world for thousands and thousands of years. Everything from corn to carrier pigeons, lab rats to racehorses, by choosing who reproduces and who continues to reproduce, we have been making all sorts of things you would not find in the natural world, but that are useful to humans. The problem, of course, for someone like me, the engineer who wants to do this in the laboratory and do it on a time scale where a PhD student could actually get a result, is that the mechanisms that nature has developed that we can use for artificial selection are somewhat limited, right? Worms with worms, monkeys with monkeys, and never the two shall meet. And mutations happen when they please, or maybe if you get a little extra radiation, you can dial in a few more, but you can't really control how the underlying DNA gets changed. You had to rely on these mechanisms. But remember, that example from Craig Venter showed us that's no longer the case. You can synthesize anything that you want. You can mutate DNA any way you want. You can dial in any number of mutations. You can choose any number of parents, 3 parents, 33 parents. Wolford here could come about by recombining DNA from different species. Think about it. This is both a blessing and a curse, right? It's a blessing because all control is off, right? You can mix DNA in any way you want. It's a curse because we have no idea what the rules are, right? We have never done this before. We have never explored what happens when you highly mutate or when you recombine DNA from different species. We don't know what kind of molecules that you will make. Now, I'll guarantee you most of them are ugly, right? They won't do anything at all. But what will you get when you have 33 parents? And this is the kind of exploration that's a lot of fun because there are a lot of really interesting things out there, and I'll share some of those with you this evening. First of all, though, let me tell you this is hard. And when I say this, it's very easy to make DNA any way you want. To have it encode something useful is the hard part, and that's where evolution comes into play. And it's hard because the space of possibilities is bigger than you can even begin to imagine. We were talking about all the different amino acids that you can now synthesize. You could increase the alphabet well beyond 20, but just think about 20 for right now. Say my particular protein that I spend a lot of my time with, a cytochrome P450, that has about 450 amino acids. If you've got 20 possibilities at every position, you have a space of possible sequences that is bigger than 80 universes of elementary particles. It is bigger than you can begin to comprehend. It's bigger than even economical numbers, bigger than the national debt. These are really big numbers. And nature has not explored but the tiniest fraction, right? Even in all those 4 billion years of all natural experiments, only a tiny fraction of those possible proteins have been made and explored. And I, in my experiments, have explored even fewer than that, teensiest fraction of that. Yet I will tell you categorically that most of them are useless. In other words, they will not solve your puny human problem. So you have a search problem. We've set out evolution works as a design algorithm, but what it is a huge search problem in enormous space that's mostly empty. So I'm going to lead you through a molecular optimization problem where you are looking in a place that's mostly devoid of function, any function at all, and you have to find something that will solve a human problem. That is the challenge. But luckily, that challenge can be solved because if it weren't the case, we wouldn't be here to talk about it. We are the products of this algorithm, and we somehow came about, and we came about for these simple reasons. I'm going to collapse a lot of data and a lot of experience onto this single slide. First of all, you can get rare functional proteins, right? Most of the space is empty, but you can get a rare functional protein by scraping it off the bottom of your shoe. They're everywhere. Functional proteins have come about through 4 billion years of evolution. They are there. Everywhere you go in the biological world, you can find folded functional proteins. So you have a great place to start. And the second thing is the landscape, the fitness landscape of this optimization problem is smooth, at least in a few dimensions. So for example, we know that most of the space is empty of function, but if you make mutations, and this is what we observe in the laboratory, you don't have to take my word for it, you can just go look at the data. If you make mutations in proteins, many are deleterious, many destroy the protein, but also many leave you with a protein that's perfectly good. We are robust, our proteins are robust to mutation. It also happens that a few of those mutations can be beneficial. They can help you adapt to changing circumstances. And what we didn't know before these experiments were actually done in the last 20 years is how incredibly adaptable we in fact are. Our molecules, our proteins are extremely adaptable, at least some of them. And that's what I've had a lot of fun playing with. So how do you do such an experiment? Now that I know that some of you have been doing cloning and mutagenesis, this is really simple stuff. You know how to do this. There is nothing complicated about this. We can take the DNA that encodes a beautiful protein, your bottom of the shoe protein, or something that might have properties that you like, but you want to confer other properties on it. So you want to make your nice corn that's richer, or your version of the protein. And you can dial in as many mutations randomly as you want, or you can recombine the DNA, and I'll tell you a little bit about that. And it's really easy to make a lot of DNA. So these are numbers, typical numbers. You can have a test tube full of DNA, all of which is different. Now, those go into cells. So you can transform the cells with this DNA. It will read it as its own, just like Craig Venter's experiment. It can read the DNA. It thinks it's its own DNA, and it will translate that into a lot of proteins. And typically, we can get cells that make 10 to the 8th different proteins. Now comes the hard part for the human being, right? You're in the lab trying to figure out which protein is better than the one you started with, because we're going to take a simple evolutionary walk uphill. We're going to find mutations that make it a little bit better than where you were before. And you have to measure that. You have to devise a way to measure the mutations in thousands of proteins and identify the cells that are expressing proteins that you like. Most of them are not. Most of them are not proteins that you like. Those go straight to the trash can in this simple evolutionary strategy. But if you have asked the right kind of question, and here's the challenge in directed evolution, it's asking the kind of question that you can solve with a single mutation, that you can get a measurable improvement in the property, and then you iterate. You feed it back into the system, and you keep improving it until it stops, till you've solved the problem or your student quits, because it's so boring. But this algorithm, this simple random uphill walk strategy works beautifully for proteins because we are the product, mostly, of such a such a strategy. And here is a great example of how directed evolution was caught by Roger Chen early on. He won the Nobel Prize for taking 2 fluorescent proteins from jellyfish, right? And he converted those by directed evolution into a whole palette of fluorescent proteins that give you all these different colors and that have completely changed the way that we visualize systems in biology. I wish I had done that. Anyway, he truly deserves this because then he went and showed how it really changes biology. Okay, so this is what I work on. I work on enzymes. And the reason I work on enzymes is they're really hard to engineer. They're really, really hard. They're complicated. And here's an enzyme. So I'm embedded in the chemistry department. You'll appreciate this. And the chemist said, oh, why don't you do something I can't do? You know, after I'd done, proteases or hydrolytic enzymes that really aren't chemically all that interesting. Their specificities are interesting, but chemically they're not. So I started working on this enzyme. And this is an enzyme that even the Caltech chemists have to respect. And they don't respect very many people. This enzyme inserts oxygen into the unactivated CH bonds of things like hydrocarbons. Think methane to methanol, right? It can insert an oxygen and stop right there, a partial burning reaction. And that is a tough chemistry to do at room temperature and atmospheric pressure in water. No human chemist has figured out how to do this, but nature's figured it out in this cytochrome P450. Now, I happen to work with one that I do really scrape off the bottom of my shoe. It comes from a soil microbe, Bacillus megaterium. And this particular version of the enzyme inserts oxygen into the subterminal position of fatty acids. Nobody, as far as I can tell, cares the least bit about subterminal hydroxylation of fatty acids. So we're not interested in this enzyme because it does that. I'm interested in this enzyme because it's well behaved and I can morph it into lots of different things. And it does its work through activating oxygen at a heme cofactor. So this is a heme monooxygenase. All right. So I'm going to collapse a whole lot of work into this one slide. A number of students over the years turned the crank on this evolutionary process and said, could we convert this fatty acid hydroxylase into something that hydroxylates small gaseous hydrocarbons? Why would you be interested in that? Once again, think of taking natural gas to a liquid fuel at room temperature and pressure. Wouldn't it be interesting if a biological system could convert gaseous alkanes into liquids? So we showed that if you started evolving this for, instead of a fatty acid, for a hydrocarbon like octane, so you can see here, the wild type enzyme, what you get from the bottom of the shoe, has no measurable activity on propane, but has a little tiny bit of activity on octane. So we could breed it by screening for activity on octane. So that's this middle bar here. And you can see that you can increase, sorry, it's this purple bar here. You can increase the activity on octane. And what happens is that along with that comes a little bit of what we call promiscuous activity on propane. So all these enzymes have side activities that aren't important in nature, but are really important for evolution. So we could start screening on propane, and after a few generations, actually a number of generations, we created an enzyme that was as good hydroxylating propane as the wild type enzyme was in a fatty acid. It became what we call P450 propane monooxygenase. It was remarkable that we could do that because people told us, oh, P450s won't take gaseous substrates, because they'd never seen one. They thought that they couldn't do it. But it's very easy. And if nature had wanted to do this, she certainly could. And probably PMOs will be found out in nature not too far from now. But let me point out that once you have the answer and sitting in your refrigerator, you can go back and reverse engineer it. You can do biochemistry. And when you do biochemistry, you can do things like stare at the mutations and see if they will tell you a story. You know what story they told me? You have no clue how these things work. There are a bunch of mutations near the active site. This is the substrate that goes into the enzyme. There are a bunch of mutations near the active site. You might even think, you're clever enough to figure out what those are doing, but nobody's been able to do that. But look at these mutations that affect the activity of the enzyme. They're lying 30 angstroms away from the active site. we can't even explain those, much less predict them. So if you're going to be the person who's going to go and write me out the DNA sequence of a P450 propane monooxygenase, I wish you good luck, because we don't even know how they work. But this enzyme is just as good, or almost as good on propane as the wild-type enzyme is on fatty acids. And it forgot that it was ever a fatty acid hydroxylase, and then the starting enzyme had no such activity. So let me summarize this first part of the talk. Proteins adapt. This and many other experiments from many other laboratories show beautifully how proteins rapidly adapt through a simple uphill walk. They adapt with just a few mutations, and the scary fact is you don't know how. So I summarize all my 20 years of experience here by saying details matter in biology, folks, especially in enzymes. And we don't understand those details. So get over it, right? Figure out how to engineer these systems without understanding the details. And evolution is exactly one of those. Let's move to sex. So I showed you one evolutionary strategy, a simple random mutation uphill walk. But nature clearly uses multiple search strategies for creating new biological objects, and one of those is recombination. Now, molecular sex is, humans have been using that, right? You know, we breed dogs this way. In the laboratory, it's not as interesting as it might sound. We do it in a fairly conservative fashion, although I can choose 33 parents, and I can mix species, but you will mostly get junk unless you do it in a conservative fashion in this way. So what we do is we take homologous proteins. Those are proteins that came from a common ancestor. And proteins that come from a common ancestor share a common structure. Their functions may be different in important ways, but their structures are similar. We find that if we recombine DNA that encodes proteins of common structure, we have a relatively high probability of creating progeny that also have that structure. So they fold. But now they have a different sequence. So they have 2 eyes, 2 ears, a nose, but they are different from their parents. And they are different in their sequences, and they are different in their functions. And how are they different? Well, let me teach you a little lesson. So sex is conservative. One of the benefits of recombination is that you can bring sequences from really different people or proteins, and when you have progeny, they will fold in function. And that shows up here. So if we look at the probability that a protein will fold as you make a number of random mutations, it drops exponentially. Mutagenesis is deleterious. But if you make chimeras by recombining 2 parents, you also drop the probability, but not nearly as fast, not nearly as drastically. So sex is very conservative. Now, we make it even more conservative. We push this probability up by contaminating the experiment with knowledge. I try to avoid doing this because usually our knowledge is not useful. But in this case, it turns out to be useful. We choose where to recombine proteins. We choose crossover sites to make progeny sequences, and we choose them based on the structure of the molecule, using a simple rule: thou shalt break the fewest interactions. And that's a rule that we understand from computer science, from genetic algorithms, leads to functional solutions. Here we know the 3-dimensional structures, and we can choose the crossover sites such that the progeny have the fewest broken contacts. When we do that, we can make a lot of new sequences. So this is an experiment we did many years ago now, where we made 3,000 new cytochrome P450s by recombining 3 sequences that differed in hundreds of amino acids. If you do that randomly, most of the sequences don't fold. But if you do it using our algorithms, you get half of them fold. And that meant out of 6,000 possibilities, we had 3,000 new cytochrome P450s. That doubled the entire functional P450 database in one experiment. Furthermore, we get things you would not find in nature. You can't find these sequences in the natural world, the not folded guys. And those are very interesting to people like computational biologists who want to test their models. But the most interesting thing about these enzymes is they're sitting in my refrigerator. They're not in a database somewhere. And because they're in my refrigerator, I can ask them, what do you do, right? I can test them. I can see what new properties they have. So I've shown you that recombination is conservative, but it's also highly innovative. And this is why sex is propagated. Otherwise, yeah, most of you in this audience would not be here, right? Come on, guys, you're not carrying the next generation. Why are males carried on in evolution, right? We'd all be having babies, all of us, if asexual evolution was beneficial compared to sexual. And the understanding, at least one of the roles of sex, is this ability to innovate at the molecular level. So we see that in P450s because we can find progeny sequences when we recombine fatty acid hydroxylase. You take 3 fatty acid hydroxylase, and I take 3 just to show that I can. You take 3 of those, recombine them, you can make enzymes that now hydroxylate or do a chemistry like you have in your liver, right? These are your liver detox enzymes that, for example, make the human metabolites of drugs. Eli Lilly gave us some drugs and said, make all the human metabolites, and we did in a single experiment using these modified bacterial enzymes. Oh, and here's another example of why sex is cool. When you recombine these 3 P450 enzymes and you measure one property, here we're measuring their thermostability, their ability to withstand high temperatures. You find that most of the progeny sequences, we're looking at the number of sequences, this histogram of all the sequences, we've tested 200, most of them look like their parents, right? Most children look like their parents. They lie within the stabilities of the 3 parents. Some are dumb, right? Some are less functional than their parents. In fact, there's a big tail out here, but I threw most of those unfolded guys away. But here's what's really interesting. Here are a bunch, about 10% of the sequences are smarter than their parents. This is the innovation aspect of evolution. We can find proteins that do things their parents never did. I showed you that on the last slide, and that are also better than their parents in a lot of physical properties. What else is cool? Engineers like this. If you have, what does an engineer do or a computational biologist do when you have a couple hundred measurements and you have the sequences of all those? You build sequence function relationships, right? So do a simple model. Imagine, you know the sequences of all these. You can do a linear regression. You can do a really simple mathematical model because there's only 8 blocks in this. We've only cut it at 8 different places, so there's only 8 elements of the sequence. There's only 3 parents. There's 24 data points. There's 24 parameters. There's 200 data points. We can build a model and predict the stabilities of every child in that library. Every possible P450 you can make by recombination. And if your model is good, you can predict, for example, the sequences of every single one that's more stable. There are 300 in the set that are possibly more stable. And when we went and made 51 of those, every single one was more stable than the parents. So you can start using machine learning tools or models to then predict where evolution can go. All right. So this is the last part of the talk. So I've been having a lot of fun making molecules have sex and optimizing proteins and spinning off lots of companies doing that in the meantime. I decided to work on the real hard problems, and that's the true innovation. It's like the species problem. How does real innovation, how does a whole new enzyme get created? Right? People say, oh, Francis, you can make a P450 more stable, or you can make it take a new substrate, big deal. They always do that to me. But how do you create something, an activity, a completely new activity that enzyme never had? Well, evolution does that, right? We know that enzymes came about through evolutionary processes. We don't really know exactly how it happens, but I believe that they branched off and created new enzymes, and there's lots of good examples in the literature with molecular level detail about how new enzymes would appear, especially ones that appear in response to challenges that humans have placed on this planet for only 50 years. Antibiotic resistance, pesticide degradation, new enzyme functions have come about as a result of those challenges. So it is the equivalent at the molecular level of how does a new species come about. And one of the important adaptation mechanisms is providing a new niche. So when you provide a new niche in your body by taking antibiotics, enzymes will adapt and evolve. So the question in the laboratory, though, if you want to do it on a time scale of a graduate student, and I guarantee you, students, if you apply to Caltech, I will try to make your project fit within a PhD of a graduate student. We try not to avoid the 400 billion year, projects. But so that translates into this simple question: how do you direct the evolution of a totally new function through a process that really happens a single mutation at a time? Unless you're a great designer and you can jump two at a time. Well, look at how the P450s did it. That's where the inspiration can come. So I'm not going to take you through this catalytic cycle because it's horrendously complicated. And but I want to point out that this is a beautifully finely tuned machine that takes its substrate, another substrate, oxygen, creates a bunch of highly reactive intermediate species before it releases its product. And nature has used every single one of these intermediates. So for example, we've got this compound 1, which is responsible for hydroxylations. We have other intermediates in the cycle that when you go out and you look at the P450s that are now in the biochemical literature, you can see that compound 1 is the mechanism, is the reactive intermediate in all of these reactions and all of these enzymes that come from different organisms. And the pink intermediates, for example, are found in this sulfoxidation reaction, or this hydroxylation, or this dehydroxylation. You've got direct nitration. Look at that cool one. That comes about from this blue intermediate. That's a really wild reaction. Nature, what happens is she recognizes these intermediates, and when a new niche becomes available where that intermediate can go to a different product and that becomes beneficial to the organism, that can come through in natural selection as a side reaction of existing enzymes. So here's a simple way of looking at it. You've got a P450. Nature has taken this and all those other intermediates and created enzymes that catalyze all these reactions by utilizing these promiscuous functions of the enzyme. And natural selection was the driving force, and a new niches in the form of biological reagents provide those new niches. If you want to create versions of, say, a cytochrome P450 that can do reactions not known in nature, what do you have to provide? Well, one might be a new niche in the form of a synthetic reagent you don't find in nature, think a pesticide or something like that, and you create new reactive intermediates. And chemists in the audience will recognize these carbenoid and nitronoid species that are well known in transition metal catalysis. So we can look at this and say nature created this intermediate. I can now force an enzyme to create those intermediates, and perhaps then with directed evolution we can do reactions that are known in chemistry. So here's the question: can you form the intermediates, and can they evolve? Can that enzyme then evolve as if you were now creating a whole new family of enzymes? So in my last few minutes, let me show you an example. Don't worry about the details of the chemistry. Take a look at this reaction. If you take styrene and ethyl diazoacetate, you can make 4 different products from that. And that drives the chemists crazy because they want to get just one product. And it's very hard to design a catalyst that would make selectively one of those products. But you feed that those reagents to an enzyme, you find that some of them that have hemes, you find some of them have no reactivity. We call this total turnover number. It's the total number of times the enzyme will turn over with that substrate. And you find that free heme, just the heme by itself, will catalyze this reaction 73 times. And all the proteins do it worse than the heme. That's not the interesting number. The interesting number is this one. My favorite P450 will do it 5 lousy times, but it will do it with selectivity for these different compounds that differs from the free heme. What does that mean? That means it's happening inside the active site of the enzyme. It's forming this intermediate, and it's catalyzing this reactivity with selectivity that's imposed by the enzyme. And you know me, 5 doesn't matter at all. 5 is a number I love because I can start from that and evolve it to become 500 or 5,000. So that's exactly what we did. Just a single mutation. Here's an example. One single mutation drove that reactivity up to 323 and made one single product. This guy here, already incredibly impressive. Here's another set of mutations that increased the activity and now made a very different product. No chemist knows how to do this with smaller molecules, but the enzyme has this selectivity. So we wanted to know, okay, here's an enzyme we've created that in vitro has these things. And if you notice, the chemist should be howling here. We've got some nasties here, nasty reagents like dithionite that aren't remotely biologically compatible. So to make this work inside the cell, so we want to create enzymes that now allow you to branch off in whole new metabolic pathways. To do that, we have to solve a very practical problem, and that is that we had to use dithionite to reduce this to the iron 2, which is the catalytically active form. And these beautifully finely tuned machines are tuned so that they won't do that. You cannot do it with endogenous reactants, reductants, unless the substrate's there. So we had to bypass this somehow, and the way we did that was to make a mutation to an amino acid that is never mutated in nature. Every single P450 that has ever been found in nature has a cysteine at that position. But we said, okay, let's make it something else, a single mutation. Then we get efficient reduction by endogenous reductants. And in fact, that makes a really nice enzyme, which is sickly green. So P450 is known because it has a peak at 450 in the carbon monoxide difference spectrum. It's a nice bright red heme protein. I've made it a sickly green. Everybody thought that was an inactive protein. It's not inactive. It just does something totally different that no one ever asked before. And this enzyme, am I the mother of a new enzyme family, by the way? Tell me. What is the definition of a whole new enzyme? I think I'm the mother of a new enzyme family. But this is now, we call it a P411. So anyway, what's cool about this enzyme is not just that it works, but it now works better than what the chemists can do. This does almost 70,000 total turnover numbers inside a dried bacterial cell. So it's just a powder. You can put it in a bottle. You can tell your chemistry friends it's a catalyst and they'll use it. If they thought it was bacteria, they would never use it. But this one they will actually use, and it works beautifully. You can do the same trick with nitronoids, and I'm not going to go into that because this talk just gets impossibly long, but we published a paper recently where we could do an intermolecular nitration, self-imidation reaction by transferring this nitronoid to this species and make these products here. And these are really interesting reactions, also with very high selectivity. So to summarize this idea, how do you create novelty? By a single-step walk is you contaminate it with some chemical intuition. That's the answer. But you utilize the inherent ability of biological systems to do more than what you think nature asks them to do. All of these enzymes can do a whole lot of things that no one ever asked. And this is the whole basis for the evolution of novel functions. And if you can recognize their potential, you can ask them the right question. And if you ask them nicely, and you make a few mutations, they will start doing things you had no idea were possible. Cyclopropionation, direct CH emanation, and many more reactions. So let me summarize. Old enzymes can do some pretty cool new things, just if you ask them. And we can use this promiscuity to move forward with evolution. And because these are already highly evolved systems, right, they are the products of evolution. Unlike rationally designed enzymes or even catalytic antibodies, which don't evolve, these evolve and you can create whole new families with them. And this is not just empty claim. You can buy these now, right? They are now available through Strem. Anybody here use Strem? You can buy these catalysts. You can buy, Buchwald catalysts, Jacobson catalysts. You can buy now my P450s because they really do work and people want to have them for these reactions. So young people, the space of possibilities for sequences is really, really big, and there's room for you out there. Biologists, this is your little space, right? This is, if you are a biochemist and you are reverse engineering biology, you are relegated to this little tiny piece of sequence space. But engineers, by making all these new molecules, we can go and explore what's possible, right? Because now we can synthesize any DNA we want. And if we explore it in a systematic fashion, that's what this is all about. How do you explore this space of sequences in a systematic fashion? You will find interesting and useful new molecules that really have no counterpart in the biological world. So let me thank some brilliant students, including Mud grad Nikki Peck. Anybody knew Nikki? Yeah, so she's now at Google. She took a year off to follow one of the postdocs to an internship doing chemistry. Biology and chemistry at Google X, but she will be back, she promises, to finish her PhD, and all these other brilliant young folks. And thank you for inviting me and for your attention.